A very good evening to all of you. I'm Dr. Vijay Paramanik from IG into Amarkanta. I'm going to present a small piece of work entitled this Bioinformatics, a gizmo to identify hotspot for aggregation in polypeptide in neurodegenerative diseases. I'm presenting it for the synthetic biology refresher course organized by HRDC RDB Jabalpur. And I'm really thankful to HRDC RDB Jabalpur for giving me the opportunity to participate in this meeting and speak in this meeting. The title of talk that is a gizmo to identify the hotspot of aggregation. So gizmo is just a tool. So here the bioinformatics will serve as a tool to identify the hotspot aggregation of proteins, polypeptides that plays a great role in neurodegenerative diseases. Bioinformatics, as you have been listening to this, as a part of synthetic biology that actually integrates evolution, behavior, animal science, computational science, biochemistry, molecular biology, pathology, and almost all field in biology and other part. So it is a complete understanding of a system by that here we can study the organism, cell, signaling network, proteins, RNA, DNA, biomolecule, environment, clinicians, and all just come together to study a particular problem, a specific problem to identify. Here, an interesting talk I'm going to give that how we are able to identify the hot spot which is responsible for neurodegenerative diseases. So we all know that protein is the functional player of life, functional player of cell. So it does the function, but what happened to that protein after doing the function? Actually, that protein is, has to be degraded, right? If it is not degraded, that is accumulated. If the accumulation of protein takes place, it will cause a number of diseases, including neurodegenerative diseases. So I'm going to discuss about that. So the next, this theme of my presentation is the bioinformatics I'm going to deal with, central dogma, proteins, protein degradation pathways, neurodegenerative diseases, and protein aggregates. Aggrescan, a software that is used to find out the hotspot responsible for neurodegenerative diseases, and then conclusion. So what is bioinformatics then? As you go for now, go for the bioinformatics terms in Oxford Dictionary, it is a branch of science that concerns with the informations and information flow. Two important things: information and information flow in biological system, especially the use of computational method. So it is a computational method to identify different prediction sort of things. So it is the application of computational tool and then approaches for expanding the use of biological, medical, behavioral health data. And when this data is stored and when it is required, you can retrieve the data from that source. So this is actually bioinformatics. What is computational biology? People always get, most of the time get confused. So what is the difference between bioinformatics and computational biology? The computational biology, is a little bit different, the development and applications of analytical and theoretical methods, mathematical modeling. And bioinformatics actually a part of the computational biology where we use the software, but the mathematical modeling and computational simulation that actually the work of computational biology. So this is a difference between bioinformatics and computational biology. So altogether, when you discuss about the bioinformatics, it is only the molecular part. Molecular bioinformatics is there. So the one dimension, you can just think about that. Only 4 digits like A, T, G, C, and 20 amino acids. These 24 players are the theme of bioinformatics. So actually in molecular bioinformatics, we study one-dimensional information of DNA, two-dimensional information of RNA, three-dimensional information of protein, and the four-dimensional when you evolving the living system. So the biologist, we can say like that. And the biologist, what the biologist did here? The biologists collect molecular data, RNA, DNA, protein sequence, and gene expression. Computational scientists here, they develop tools to analyze that, software, algorithm, to store and analyze the data. So the bioinformaticians are, we study biological questions by analyzing molecular data. Here our job is to identify neurological disorder by that. It is a very useful and a very, very principal slide, we can say like that. The central dogma. DNA makes the RNA that makes protein, right? So it is known to all. They create this DNA, and from DNA is just a script writer of the movie, and that each codes the RNA messenger is made, and from messenger the protein is made. Protein does the function, but after doing that function, that protein has to be degraded. If the protein is not degraded, it is accumulated in the system, in the cell, and ultimately causes disease, including neurodegenerative diseases, right? So this is eukaryotic system, and in prokaryotic system, this transcription translation takes place simultaneously in prokaryotes. In eukaryotic system, there is a different stepwise method. So it is a simple, this is all it is known to all of us, that explains step protein synthesis. The messenger is made from RNA. The protein is made. How this in the ribosome, and after the protein is made, that is chaperones comes, then and a folding takes place. Then it is sorting of the protein takes place. It goes to nucleus, mitochondria, membrane, whatever the function is required. And after doing that function, the protein has to be degraded. Amino acid sequence already known to that. Amino acids that with electrically charged, like positively, negatively. Then special special cases like cysteine, glycine, proline. Amino acid hydrophobic side chains. There are some aromatic amino acids like phenylalanine, tryptophan, and tyrosines. We have 3-word code and 1-word code. This 1-word code is one letter code rather. One letter code is very important for the bioinformatics. So we just alanine to that's A, just like that. It is no need to speak like that, and it's all known to all of you, right? So the protein degradation, what I'm telling, the protein degradation is one of the most important pathways, ubiquitin proteasome pathway, right? Important because after doing the function, protein has to be degraded. So 2 important pathway that takes place for the degradation of protein. One is ubiquitin proteasome pathway. 80 to 90% of the proteins are degraded by this pathway. Most intracellular proteins are there. Lysosomal processes also degrade the protein like 10 to 20%. In this category, the extracellular proteins are degraded. Cell organelles, some intracellular proteins are also degraded. Protein degradation pathway that takes place by this ubiquitin proteasome system, UPS system, like carboprotein is there, and proteasome is a big complex molecule, and in this protein, the protein is degraded after doing the function. One more degradation pathway is there, the autophagy. Autophagy is like macroautophagy, then chaperone-mediated autophagy, then microautophagy. There are 3 types of autophagy. This is also responsible for protein degradation, but the most integrated system, most important protein degradation pathway is ubiquitin pathway. So how, when the function has to be done, this message is made, protein is made, right? And from protein has to do the function, but after doing the function, protein has to be degraded. When protein has to degrade, what is the pathway? That is the important aspect. And the ubiquitin is a monomer, ubiquitin molecule, that ubiquitination of the protein takes place. And after ubiquitination, once the ubiquitination takes place, the protein has to be degraded by 26S proteasome complex. So this is activation, ubiquitin activation is required. Then ubiquitin conjugation takes place. And once this actually the conjugation is there, then they are target the protein. Target protein is identified by isoamide bond, and when it is recognized, once this tagging of this protein ubiquitination takes place of any protein has to be degraded, and it is very useful for the system, for the cell. If the protein somehow is unable, or the proteasome complex is unable to identify the protein, then what will happen? There is accumulation of the protein. An accumulation of protein takes place, then there is neurodegenerative diseases in the brain. So the protein aggregation in neurodegenerative diseases, you know that aggregation mechanism, there is monomers, then its dimer takes place, then oligomerization takes place, then finally, then aggregate takes place. This is from decalcitonin. Like Alzheimer's disease, amyloid beta accumulation takes place, tau in Alzheimer's disease, Parkinson's disease, alpha-synuclein, then ALS, the FUS protein the aggregation of FUS takes place, ALS again, TDP43, like ST Huntington disease also, the STT protein accumulation takes place, right? So this is a very exceptionally well paper that actually gives a very good gizmo tool to identify the protein, hot spots in the protein molecule, whether this protein has a potential to be accumulated in the system. So the protein aggregation and the neurodegenerative diseases by Ross and Ross, Ross and Missel. That is a mind-blowing paper that suggests and that is a bit like that. So Huntington disease, the Huntington I already told, Alzheimer's disease, the sporadic, and then Parkinson disease, the ALS, and the Prime's disease. These are the neurodegenerative diseases, and most of the diseases are due to the accumulation of some of the protein, either misfolded proteins or those proteins which are not recognized by proteasome pathway, and the proteins are not degraded, and actually they are accumulated. So this is one of the paper. Then I will take the reference of this paper, the prediction of hotspot of aggregation in disease like polypeptide, right? So BMC structural biology, this is published. So the software I'm going to use in the Aggrescan all in capital. So what I have to do, like suppose you take in proteins, the ApoE proteins, which are supposed to play a role in in disease conditions. The ApoE, PS1, PS2, and these are the some of the signature proteins which are responsible for the clearance and are responsible for other Alzheimer's disease. So ApoE proteins will go to the this NCPI, then try to find it if we human, then we'll get the faster sequence like that, as we'll already learned in this course, and then this C prompt NP protein sequence will be found, and then what happened, they'll have subjected this protein sequence into the Aggrescan software, so that will find out the hotspot of this in ApoE protein. So when they are subjected to this scan, they will find there are 5 number of hotspots, 5 things normalized. These are the sum of the formula that we are focusing on that. The hotspots number of hotspots are 5 in this ApoE protein. So this is very important to learn. These hotspots number, the higher the number of hotspots, that means the chances of aggregation of this protein in this system on a cell or in the brain is high. Right, so there is some calculations on that. I'm going into the detail of that. So the hot spots, this is regions with 5 or more residue. The frequency with, there is some calculation on that, like that is responsible up to stress of 10 amino acids. So which are actually predicts the number of hot spots. If the number of hot spot is high, that means the chances of aggregation of that protein is very, very high. So the number of hotspots is identified. So this, it's a very small sort of thing. So when it is identified, the hotspots are identified. So we can be able to find out whether this protein, in therapeutics potential of that proteins, either these proteins has, which of the proteins, this has the less number of hotspots. Less number of spots, that means that are not accumulated. If we take the Huntington proteins, the hotspot will be very, very high. In the top proteins, the hotspot of the protein is very high. So somehow you can change the sequence of the protein, change the sequence of the protein by in the wet experiment to find out and to decrease the hotspot of that. So can we, neurodegeneration of the transition takes place very little. Okay, so the bioinformatics, the Aggrescan is a tool that can give information about the aggregation potential of protein on the basis of amino acid sequence. This is helpful to carry out the weight experiment and designing the therapeutic molecule, just like weather forecast, So able to know, but we have to prove by experimental basis. So with the data which is subjected by this Aggrescan will provide you the number of hotspots. So with that, this is a small part of that. Let us wrap up in that. The protein is a functional pair of a cell or a life, you can say like that. But after doing the function, the protein has to be degraded. It is degraded by a pathway that is called proteasome pathway. That is the main pathway in the eukaryotic system. Other is autophagy by mitochondria. This protein, after doing the function, this protein had to be degraded by proteasome complex, right? So if it is not recognized by ubiquitination, so ubiquitination by ubiquitin enzyme, So it is not degraded. It is not degraded. That means the neuro and accumulation of that protein takes place, and accumulation of that protein means there will be neurodegenerative diseases. So that potential, a particular proteins, that hot spot for the aggregation potential can be identified by using a synthetic biology software, synthetic biology approach, that is by bioinformatics tool, that is Aggrescan. So Aggrescan will be able to find out the aggregation potential, right? Aggregate potential of a particular protein. So that will be just like in forecast, where that forecast we are able to know what is the potential of that. So with that, I end the slide, and I welcome all of you, the participant and organizer, to welcome to Argent Trauma Contact, and with regards. Thank you very much.